will be Dr. Anthony Fitzpatrick, and he'll be giving us a short presentation on the Cryo-Electron Microscope Center at the Cancer Center. And Dr. Fitzpatrick has a PhD in biophysics from University of Cambridge, and he also completed his postdoctoral training at MIT, Caltech, and at Cambridge, and he's currently an assistant professor of biochemistry and molecular biophysics at the Zuckerman Behavior Institute, and he's a PI of our HICCC Cryo-EM Microscope Center. So Dr. Fitzpatrick, are you ready to give your presentation?
Yes, I think you should see the first slide, right? You all see it?
Yes, we can see your first slide. Thank you so much.
Okay, thank you so much for the introduction as well. So I'll be talking in the second half of today's research seminar about cryo-electron microscopy, which is currently the highest resolution technique to visualize either proteins or protein complexes, protein RNA, protein DNA complexes, or even cells at highest resolution possible. And I think this is really exemplified by what we have been able to do just as a sort of test case with a standard protein on one of the cryo-electron microscopes here at Columbia. So what you're seeing in this first slide is just every single amino acid that we have been able to resolve almost to atomic detail on one of these cryo-electron microscopes. And I think a couple of the key numbers are, if you can get down to a resolution of 4 angstroms or below, you start to resolve individual amino acids in your protein complex. And if you can get down past, say, 2 angstroms, you're starting to get into the range where you're starting to see individual atoms. And this is incredibly powerful because you are able to solve the structures of unknown protein complexes, of which there are many important macromolecular machines that we do not know the structure of still, even though structural biology has a history of 50 years, crystallography and NMR have not been able to solve these structures. And I think Cryo-EM is, since about 2015, when we underwent what was called a resolution revolution, which really brought us down from about, say, 1 nanometer, which is 10 angstroms, which was kind of a hard limit for us at that time, all the way down to single amino acids and now also single atoms. And Columbia have invested heavily in this technique through the help of the Irving Foundation and other gifts. And I think this was also really a standout success for Columbia with the award of the 2017 Nobel Prize in Chemistry to Dr. Joachim Frank, who was awarded the prize for his development of how to figure out the image processing techniques that we need so that we can take images of protein complexes and actually generate high resolution 3-dimensional volumes from that set of images. So the basic premise of what's called cryo-electron microscopy single particle analysis, which is mostly what's happening at Columbia currently, is that you want to take a protein complex, so just this sort of gray shape up here, this gray volume. This is a Cryo-EM macromolecular machine. And if we image this on a cryo-electron microscope shown here on the right, we actually can generate 2D projection views of this protein complex. And since the machine itself is viewed at different angles when it is being imaged, you can actually put together those 2-dimensional images, knowing the angles that they were taken at, into a high resolution 3-dimensional volume. And this is incredibly powerful because unlike X-ray crystallography, we don't need to form crystals. We also can use very small amounts of protein. So typically the sample that we use shown here, we would typically need about 3 microliters of protein at around a concentration of 0.5 to 1 mg per mil. This sample is then vitreously frozen onto a grid. This grid is about 3 millimeters in size. And we plunge freeze this into liquid ethane so that we can quench the system, quench the buffer, so that it doesn't form sort of cubic or hexagonal ice, but instead forms this vitreous ice, which is where the Cryo-EM comes from. And this really preserves the shape and structure of the underlying protein complex that we've purified, so that we can directly visualize it in one of these Titan Krios microscopes that we have as part of the core here at Columbia. And then from these images, we take sort of tens of thousands of images of the protein complex, all in different orientations. Then we align self-similar views. So here you're seeing sort of a square, circle, triangle. And we do this to increase the signal to noise of each of these different views. And by then determining the angles at which those different views represent, we can actually create a three-dimensional density map of the protein complex. And then from that, if we're at sufficient resolution, we can create a three-dimensional protein structure from that. And the core here at Columbia has 4 of these Titan Krios microscopes, which are currently the most powerful microscopes that are on the market. They're about 16 feet in height, so they're incredibly sort of large and complicated to operate. And there is a great deal of assisted usage through the directorship of Art Palmer, who is the director of this core, and also the day-to-day operations are done by Bob Grasucci and his team. And we also have a second smaller microscope called the Glacios, which is really good for screening your grids of your samples that you may be interested in collecting higher resolution data on the Krios. This is a slide shared by Dr. Chi-Min Ho here at Columbia. I think it really captures the way in which Dr. Joachim Frank was able to put together these two-dimensional views using image processing to create a three-dimensional volume. So if we play this again, what Dr. Ho has done here is she's collected many, many images. Those views of the particle are then sort of separated into different side views, top views, oblique views. And by doing so, you can actually recreate the three-dimensional volume of the protein complex that you were trying to solve. And in this case, what I think is really nice about this complex is that this is a really important drug target in malaria. It's actually a protein complex that is responsible for actually the export of many of the proteins that cause malaria once the parasite has invaded the body. And so everything goes through this remarkable machine called PTEX. And the exciting thing about Cryo-EM is that not only can we work with purified recombinant samples that we've overexpressed in cells and then purified, we can also pull things out directly, as Dr. Ho did here, from living organisms. So in my case, I actually do this from human brain tissue. So we can also solve the structures for the first time of endogenously sourced protein complexes. Some other examples of some very exciting work done using the Cryo-EM core here at Columbia are shown on this slide. So the Shapiro lab has done very important work with Dr. David Ho on COVID and the spike protein. The GABA B receptor by Ching Fan, Dr. Fan's group solved this very important receptor. Also, Dr. Mancha with most recently a omega-3 transporter. Dr. Sobolevsky has done very important work on glutamate receptors. And also Liang Tang's group have done a real range of structures using the core of Cryo-EM here at Columbia. And then finally, the ryanodine receptor, which is a huge, important macromolecular machine, primarily by Dr. Marks and Dr. Hendrickson and Dr. Frank. As I said, one of the exciting things that I find about Cryo-EM is the prospect of being able to solve protein complexes purified from actual endogenous sources. And so my interest lies in sort of neurodegeneration and neurodegenerative diseases. And so I have been able to purify from human postmortem tissue filaments that are associated with neuronal cell death. So here we take an Alzheimer's patient, a few grams of tissue, and we purify out a filament formed by the tau protein, which is shown here in an image. You see these halos. Those are halos of tau filaments built up inside neurons, clogging the neurons and contributing to cell death. We've characterized them further here using antibody staining. And when we purify them out, you'll notice that there are two types of filaments shown here in blue and green. And the sample is not particularly clean. You can also see some other junk here, like this is apoferritin. There's some other sort of molecules around. But the power of Cryo-EM is that we can take different views of just these filaments and actually solve their three-dimensional structure, cut through that density map, and actually find the underlying sequence underneath. So we can actually build into the density map the native structure of the protein forming these toxic protein aggregates. And so just a quick sort of demonstration of how this works for my area of research. So these filaments, first of all, you start off with a low resolution map. You then start to see individual molecules, individual beta strands. And when we turn this 90 degrees and we cut through, and this would be the same for any of the other examples I showed earlier of single particle analysis, you really start to see a density map in which those amino acids are so well resolved, you can actually build the amino acid sequence directly into your Cryo-EM map. And I think this is an incredibly powerful technique that still has a lot of discovery and mileage to go in terms of solving important protein complexes. So you can see here, these little bumps are actually real sort of side chain densities. And the different sizes of those side chain densities allow us to build a amino acid model of the protein directly into the Cryo-EM map. Now, today I was also asked to talk about the future of this field and where we might all be headed. And one thing that is important about these sorts of endogenous sources is, well, could we actually, instead of purifying out the filaments or any other protein complex that we're interested in, is there a way that we could, instead of just solving these structures to high resolution by purifying them out, could we actually look at them in their native environment? So here, this is a neuron, and I'm showing an artist's impression of where these filaments would be and what they might be doing. But what we really want is in-cell structural biology. We would love to be able to solve structures directly in their place of work and in their place of function, which is in cells. And to do that, we will all have to start doing what's known as cryo-electron tomography. And this is a similar principle to single particle analysis, but instead of purifying proteins, we actually freeze whole cells or even tissue onto these 3mm grids that we plunge freeze again into liquid ethane. We use our Krios microscope again, which is part of the core. And instead now we're taking images of those cells at different angles. And by creating these different angle views of the cell, we can actually generate a three-dimensional reconstruction of the cell purely from these 2D projections. And I think that this is a bit more involved. My lab has been able to put together most of the sort of hardware that you need to do this. Firstly, you have to vitrify the sample, which for cells can be more involved because they're much bigger than protein complexes. They may require certain tricks in order to freeze them properly. The other thing is that an electron beam can only pass through about 0.3 micron of material, soft material rather. And so we also have to sometimes shave down these cells, whether it's a cancer cell or a neuron. You'll have to shave it down so that you can actually get an electron beam through that portion of the cell, generate an image, and then through tilting the stage now, you're actually going to generate different views of that cell. And then finally, a lot of this work, whether it's single particle analysis or tomography, involves a lot of computational work on the back end to actually generate the reconstructions. And so this is what my setup looks like. Firstly, you start off by imaging your cells using a regular light microscope. The only difference is that everything now has to be kept at cryogenic temperatures so that you don't melt the ice. And so we have it connected to a liquid nitrogen dewar here. So everything is being imaged under cryo conditions. We then shave down the cell using this second electron microscope, known as a Fitzpatrick. And then we image in the Krios microscope at different tilt angles. And then finally, we solve the structure of the entire cell that we're looking at using a GPU cluster. And so this is just a little bit about how we do this. We are currently doing cultured neurons that we take from mouse embryos and really culture them onto these 3mm grids, vitrify them. And the reason why it's necessary to have a fluorescence component is that if you look at the cells, typically using electrons, you don't really see very much. So what we have to do is we have to use some kind of fluorescent staining to actually identify where the cell is. And then finally, when we take it through to the focused ion beam scanning electron microscope for shaving it down, we have to match the coordinates, because as you can see, it's very obvious that this is a neuron in this image, not so obvious now in grayscale. And so once we go into here, we have to match coordinates and then shave down the neurons so that we can actually know that we're sort of paring down in the right location. This is just a brief summary of what this all looks like. So we're actually preparing what's known as lamella, which is kind of a window into the cell. This is the main microscope that's used to do this. It's more of a sample prep microscope rather than a high resolution imaging microscope. But really, we put in the cell first, which is still under cryogenic conditions. We coat the cell with a protectant. We image it using an electron beam. And then we tilt the whole cell and the whole grid to an angle such that we can actually shave both above and below the cell and generate this lamella or lamellae, plural, of sort of a window into the cell. So this is still frozen, still hydrated. We believe it's still happy, despite, and these additional steps are really to protect from radiation damage. We're trying to ensure that this frozen cell is as protected as possible. We then shave this down, and we're left with a sort of thin section of the cell, which we then take across into the Krios microscope to image at different tilt angles. And you'll see that the results from this are very compelling, very exciting, because you start to see not just the structure of an individual protein complex that you would get from single particle analysis, you really start to see what's called the molecular sociology of the cell. So who is interacting with whom is talking to whom in terms of protein complexes in the cell as they would have been when you first froze that cell. So this is now your coated lamella. You're then going to take it across, transfer it to the Krios, and then finally, as I said, the way we do it here, because you only have one cell, you don't have tens of thousands of copies of the proteins like you do in single particle, is you're tilting the stage to generate a series of two-dimensional projections of the cell. And then from those 2D projections, you're actually generating something like this, which looks like a kind of artist's impression of the cell, but is actually the nuclear periphery. So this is actually just on the edge of the nucleus. You can see the nuclear pore complex here. You can see actin, you can see microtubules, you can see ribosomes clearly resolved. And so this is really kind of where the field is going and where we hope to be able to make major inroads here at Columbia using these core instruments. So this is finally what a FIBSEM can do, the lamella, the sort of window into the cell, I suppose. Really, you're shaving above and below, like I just showed. You're trying to generate a thin section that is around 200 nanometers thick. Depending on the magnification, you're getting a view of about 0.5 micron in X and Y, and then you're getting this thin sliver of the cell. And I think that part of the art here is to figure out exactly where you are in the Z height so that you know what you're actually imaging. These are some results from my lab that we've generated. So what you're seeing here on the left is the tilt series of a stage on which there were neuronal cells. You can see a lot of detail already just by eye. You can kind of see that these are ribosomes. There's some vesicles around, some kind of membranes. And whenever we do the reconstruction of this part of the cell, you can actually see now that indeed there are microtubules, vesicles perhaps being moved along on those. Purple is the ribosomes, and then this green is kind of membrane. Just another quick example. What's striking is really the contrast. You really do have good contrast for just a single tilt series. But the real challenge here has been to get the resolution of these cellular reconstructions down to the same level as we would have for single protein complexes. And then finally, just this third one here, this is really striking because the contrast is even better. You can clearly see as we tilt through the cell on the stage, we can see these microtubules, you can see lots of polysomes, some vesicles that are being probably trafficked along the microtubules. And when you do the reconstruction in 3D, you can actually infer a lot of this. You can see what's happening in that part of the cell. And now you're also able to see how close things are in terms of Z height as well. A recent proof of concept by Dmitry Teganov showed that, in fact, there is no intrinsic limit to tomography. We can, in fact, generate just as high resolution. This is a ribosome that was collected through tomographic data collection in a cell. And as you can see here, it is around 3.4 angstroms at best, which is typically the resolution we need for single amino acid resolution. They were even able to see a single drug molecule buried in the core of the ribosome. So there is no real physical reason why we cannot do high resolution reconstructions of protein complexes in the cell, which will definitely provide more information than those purified recombinantly. That doesn't stop there. I think the most recent thing is, can we actually do this instead of just with cells? Can we also do this with unstained tissue? Of course, electron microscopy of stained tissue has happened for years, decades, but there are some artifacts associated with that. And so Cryo-EM now is poised to try to solve structures of protein complexes actually within organisms. The only challenge is that when you freeze the organism, it's a much bigger block because it's here we have C. elegans buried in the ice, and we have to identify them, as you can see, because we can't see them currently, using fluorescence microscopy first. So you start to see the GFP tagged C. elegans. Then they focus in on the intestine of the worm. They then create a lamella. Now, in this case, the lamella cannot be imaged because it's buried in a huge block of tissue and ice. And so it actually has to be physically lifted out using cryo-cooled tweezers and then thinned down later so that you can actually then image this in a Krios microscope. And so this is where we're headed also in terms of human brain tissue, organoids, things of that nature. But it is much more involved because you have to, as I say, not just freeze and identify where you are, but also somehow lift out this section, very small section of the tissue, and actually then pare it down using your FibSem, as it's shown here. You're actually thinning it down here, and then actually imaging this in the Krios. I did say the contrast was pretty good in conventional cryo-EM. It could be better. We think it could be better. My lab recently got a Chan Zuckerberg Initiative grant in order to pursue this idea. One idea is that much like a Zernike phase plate, if you have some kind of post-specimen phase plate that would actually change the phase of the post-specimen beam, you would actually be able to enhance contrast And so you would actually be able to see things more clearly. And one thing that we're going to try to do at Zuckerman is to build such a microscope where you're actually deflecting the post-specimen electron beam using a pulsed laser beam. And in order to do that, the electron beam itself has to be pulsed. And you can see here the demonstration of this. So the idea is very simple. You're going to try and take an incident post-specimen electron beam and change the phase of this electron packet using a pulsed laser beam. And in so doing, you induce a phase shift of the beam, and that should actually enhance the contrast of your image. This has already been very elegantly solved by a group at Berkeley. Holger Muller showed that for a continuous laser, this can actually work pretty well. So you see here on the left a typical Cryo-EM image of a 20S proteasome, and you can see that the contrast, you can see it's recognizable, but the contrast is not very good. And with their continuous laser phase plate, they actually were able to boost the contrast quite significantly. What is hard about this approach is that you need a very high sort of fluence in order to achieve that 90 degrees phase shift you would like. And we're hoping that the pulse peak power of a pulsed ultrafast laser, which can be many orders of magnitude higher than the average power of a continuous laser, would make this more feasible and perhaps have less artifacts in the resulting image. And so that's really it from me. I think that if any of you are interested in using the Cryo-EM Quart Columbia, I think Dr. Palmer and also Bob Grasucci are the best people to contact. And also it's fully available in iLab, as Peter mentioned. Pricing and all those sorts of things are all there in iLab very clearly. So if you have any questions, thank you for your attention.
Thank you so much, Dr. Fitzpatrick. We do have one question in the chat. Have you or others added 3D tags to identify individual proteins within complexes?
Yes, so I think actually that's a very good point. Going back to the example I showed here, the endogenous complex I showed from Dr. Ho's lab, they actually used a CRISPR tag. They engineered a tag, I think it was a flag tag, in order to be able to pull out this very unwieldy complex from the parasites, because this particular protein complex never really was expressible using recombinant methods. And so they engineered a CRISPR tag to be able to pull that out. So yes, and I think that for a lot of the other examples I showed in the following slide, that is typically the way that you would try to use a tag to purify out the protein complexes.
Okay. And we have another question. What would be the advantages of the pulsed laser phase plate over the existing Volta phase plate?
Right. So I think the Volta phase plate kind of came out around 2014, 2015. And initially there was a lot of excitement because you do get higher contrast from these phase plate images. The trouble with using a material phase plate is that it really is just a piece of carbon that you insert post-specimen, and then you heat it up slightly so that you get a kind of electron cloud emanating from that. And that is what is deflecting your electron beam and causing the phase shift. The problem with it is that it changes over time. You have no real control of what the phase shift is. And I think that the promise of either a continuous laser phase plate or a pulse laser phase plate is that it's no longer a material substance. You're now deflecting the beam using a highly tunable laser, which will remain constant over days. And so you should have, in principle, a constant phase shift that could last for as long as you want. Yeah.
Okay. And we have a follow-up to that question. Do you think the laser phase plate offers the most benefit for single particle cryo-EM or in situ cryo-ET, or both?
Right. So that's why we pitched it for Cryo-ET. I think that for single particle, even the voltaphase plate, which was just mentioned, really it has now been demonstrated sufficiently that really for single particle, you probably don't need a phase plate. The information is already there. And in fact, there isn't really much of a gain to be had, perhaps with small proteins, because you can perhaps infer the orientation better using a phase plate. But I think the big gains will be made in tomography because it's so dense. You have so many proteins in these cellular tomograms. Getting any additional contrast will help you to identify the non-obvious objects. So in some of my tomograms, you see microtubules, you see ribosomes, you see vesicles, but what you really are interested in looking at is the non-obvious sort of protein complexes and what they're doing. And to do that, you will need better contrast. Yeah.
Thank you. Any additional questions? You can unmute yourself or include a question in the chat. Okay. Thank you so much, Dr. Fitzpatrick, for that informative seminar. And thank you all for attending. And you will have a survey to fill out later on by email. We'd appreciate it if you take the time to fill out those questions. And thank you all for attending, and we'll see you again soon.
Thank you.
Thanks.
Thank you.
You're welcome. Thank you for coming.